Now here in red is what's important for you to know. You need to know that this is a typical C3 green leaf. That's the majority of the plants that you're going to come into contact with. They're going to be C3 green plants that do photosynthesis using using the C3 method. There is a C4 and CAM photosynthesis that we will talk about later, just touch on. But just know that C3 is pretty much the way most of the plants that you're going to come into contact with are going to be doing photosynthesis. But when you're dealing with the leaf, as far as the anatomy of the leaf, the two main ideas you need to get into your head, and it's all written here in red, is the stoma. First of all, that you understand the stoma opens and closes, allowing gases and water to leave. Water does not enter through the stoma, but water will leave through in the gas phase. You'll notice that there's a lot of spaces here in the leaf. Those spaces allow for gas to flow quickly, diffuse through the cell membranes, letting oxygen out and carbon dioxide in. You'll also notice that the chloroplasts are layered one on top of the other. So when a beam of light comes flying into the leaf, there's a high degree of probability that this photon is going to come into contact with a chloroplast. And that's what you want if you want to do photosynthesis. So, the anatomy, the name of the bundle, sheath cell, the spongy mesophyll cell, the lower epidermis, the upper epidermis, the palisade, and the stoma, although fairly simple in my opinion, to learn, is not the emphasis of this class. The emphasis of the class is that you understand that the general design of the leaf functions as the perfect photosynthesis machine. What you have is, in general, you're going to have a plant that's going to have a kind of a broad leaf. Most plants are going to try to make their leaves as broad as they can. They're going to have all kinds of veins that go through the plant. And the broader the leaf, the more surface area you have. And what that means is that there's more of a probability, again, that light is going to hit this area. Throughout this leaf, the surface, as a light beam comes in and hits the leaf, because you have a larger space, there's a larger probability that light's going to hit it. You have rate of diffusion of gases increases because it's flat. If it were a ball or a giant mass like an apple or pear, then it couldn't function very well as a leaf because the gases would not be able to diffuse easily from the center of the mass. So if you have this giant, spherical leaf that's thick, it's difficult for a molecule of oxygen in the center to diffuse outward. Where in this leaf, a molecule of gas here could easily move outward, out of that leaf. The rate of water that moves from the soil is increased because you get the blood, these veins allow for the fluid to move up into the leaf, and as transpiration occurs, as water evaporates from the leaf, out goes the water through the stoma, creating a negative pressure that pulls the water up from the roots. Again, because we stack the chloroplasts, there's a higher probability that the chloroplasts are going to get hit by this ray of light coming down. So all these pieces of the anatomy of the leaf are very important for the functioning of the leaf. And of course, when you start looking at evergreen trees and etc., there's different advantages of those kind of leaves. They're able to do photosynthesis much slower. Their photosynthesis happens at a much slower rate, so they don't grow as quickly, but they're able to do it all throughout the winter because they're not losing their leaves every season. Okay, so again, it's kind of four major ideas you need to know for the exam. Is that the leaves are the main photosynthetic organ of the plant, that there's large surface area allows them to capture light quickly and allow for quick diffusion of gases throughout, out of the plant, but also into the plant, oxygen out, carbon dioxide in. You have an arrangement of chloroplasts and leaves that maximizes the harvesting potential. So you see the chloroplasts are stacked one on top of the other. You see the light being coming in, and even if it misses the first few, eventually, hopefully, like it's most likely it's going to hit one of the chloroplasts before it leaves out and get captured. That light photon will be captured. The main tissue involved in photosynthesis is the Palisade mesophyll tissue. And that just the idea that there's this mesophyll tissue that has all these chloroplasts stacked one on top of the other. Those are the four kind of big ideas you got to get out of this. But there's other kind of interesting points here, and that's that leaves. You have to remember that leaves are connected to the rest of the plant. That they're part of a complicated circulatory system, just like we have a circulatory system, just like the turtles you dissected have a circulatory system. So do plants. Maybe circulatory is not a good word for it, because maybe you should call it more of a transportation system. But in any case, the liquids do move from its roots up into its upper leaves. You got to remember that everything in the plant is designed to try to get this photosynthesis to work extremely well. And it's interesting that plants that grow up in dark areas where they're very shaded have more chloroplasts. So because they have more chloroplasts, they tend to be really dark. So you see the jungle plants, if you ever noticed that in movies, the jungle plants are really a deep, deep green. That's because they tend to be in shaded areas underneath other shaded plants. Where plants are generally directly in the sunlight, like some of our trees in your neighborhood, are going to have a less deep green look to them because they're going to have less chloroplasts. Very interesting. But again, the main ideas here are here listed in red. And some the connections we mentioned before, which is the xylem, or the vessels that conduct water from the soil, go to the leaf, and transpiration, in other words, evaporation of the water through the stoma, allow the water to be pulled up from the roots. And of course, as I mentioned before, the leaves are connected to the rest of the plant by a complicated and organized transport system. Now, this material is repeated. I must say, and we must have said it 3 or 4 times, everything on this page, but it's important that you do realize that this stuff is going to be on the test. And where we come to repeating is this idea here that chloroplasts are, in fact, endosymbiotic. We believe them to be endosymbiotic organisms, or at some point we're endosymbiotic. They're the remnants of a symbiotic relationship, right? So they're what's left over after the organism no longer is a self-stating organism, but still has that double membrane structure that we discussed in class, that I've discussed in a previous video. Then we look at its own DNA, its own ribosomes. So it's something that you should think about. Please go ahead and read this section in green and emphasize it, know it well, understand it. If you understand it, great. If you have questions, be ready to ask them on Monday. You have here the arrangement of Palisade cells. Again, this is something that's been repeated throughout this the whole workbook, is that these arrangements of these cells and the arrangements of everything in this plant, in this leaf, is to maximize the capturing of that light. So when you look at something like here's the stroma. Now this is stroma, S-T-R-O-M-A. That's different from stoma. Stoma is the opening in the leaf that allows gases in and out. The stroma is the space here. I know I disagree with the naming of something like that. I always feel that names should be quite different, but they're not in this case. So you'll have to just get used to that extra R as being a completely different space. We've gone through these structures before, inner membrane, outer membrane. We've looked at this in the mitochondria. We've looked at this in the chloroplast and other videos. These are the thylakoids, the stacks within the chloroplast that contain the pigments of chlorophyll, the photosystems that allow for the capturing of light. How are they adapted? This is a good question. How are chloroplasts adapted to capture light? Here you have a lot of the stuff here listed here, and some of the ideas that you might want to write about that, and we'll discuss that in class on Monday. Try to think about, as you look at this structure, what kind of adaptations do you think that this chloroplast, as you see these stacks of thylakoids, they're stacks inside a chloroplast that then is stacked again in a plant. why would a chloroplast do that? And then look at this question: how are chloroplasts adapted for light capture? And come up with some kind of an explanation. Write it down in your binder, and let's discuss that on Monday. How this might be, how this structure here is adapted to best capture light. So here you have Photosystem 2 and Photosystem 1. We've gone through these before in previous videos, so I've given it to you in videos in this workbook. Hopefully you're paying attention to it. There really isn't much more for me to say other than what you're reading here and what you've seen in the videos previous to this. Here again is a little more detail of looking at how here. Remember that question I asked before is how is a chloroplast best adapted to best capture light? Go ahead and take a look at these and read the descriptions and think about that question. And here's another question: what is a photosystem, and how does it increase the light harvesting ability of a chloroplast? So here's some description of that to answer that, and try to understand it. So when we talk about it in class, you're best prepared to do your best on the exam. So here's three main ideas. Make sure you know them. And the blue is interesting. Magnesium is important in plants. And for chlorophyll, this is extremely important. These three ideas, these will definitely be on the test. This might be on an extra credit question. I'm not sure yet. But these three main ideas have been hit over and over again. You can bet your bottom dollar that those are going to be on the exam. So understand them again, prepare your questions for Monday. Now let's go over these answers real quick. List 3 ways leaves are adapted for their job. I mentioned them in the video. I'm not going to answer that, so go back and rewind the video and look at it if you didn't get it. Also look at the previous videos that I've sent. I've also mentioned it there. Draw a simple two-dimensional diagram of cellular structure of a typical leaf. Label the diagram. Now, that diagram is in the workbook itself, so you should be able to draw that without my help. What is the job of the plant vascular system in photosynthesis? The vascular system is the phylum and the xylem tubing in the veins of the plant. Go back and look at my other videos, the 2 other videos I sent you. I mentioned xylem and phloem. They discuss it here in the workbook as well. Again, I don't think it's valid for me to go ahead and give you those answers since they are pretty obvious. Just look at those other videos or go back in this workbook and find them. Think about them and think about how that works and compare them to our own vascular system. How is the arrangement of chloroplasts in leaves designed to maximize light capture? I actually said that in class. I read it to you, or not in class, but I read it to you here in this video. So you should be able to easily answer that one. In what way do chloroplasts indicate the existence of a cell cytoskeleton? That's mentioned here. I'll give you a clue. It has to do with the evidence that the cytoskeleton is important in photosynthesis is in the blending experiment that the workbook mentions, taking leaves and blending them in a blender. Draw a simple diagram for the internal structure of chloroplasts. Again, you can find that in the workbook. I'm not going to draw that for you again. On the diagram 6, indicate clearly where the 2 stages of photosynthesis occur. You can do that. You can identify where photosynthesis is occurring in any diagram. The 2 stages. Remember, what are the 2 stages? I don't even want to mention that because, again, if you don't know it by now, you haven't been doing your diligence. You haven't given your due diligence. So go back and watch the videos and take it down. You know what the 2 stages of photosynthesis are. You should be able to diagram them in some type of leaf and tell me where is it happening. Is it happening in the stroma? Is it happening in the intermembrane space? Is it happening in the thylakoids? Where is it happening? What is a photosystem? Again, answered directly in the workbook. It's not something that I feel I need to explain to you here. How is the arrangement of enzymes, electrons, and carriers in photosystems designed to maximize the capture of light? This is a little more subtle. You need to think about how how are the enzymes positioned? Think about the leaf structure that we've discussed earlier. Think about the thylakoids and how they're stacked up one on top of the other. Think about what light is. Think about light coming in through your window, for instance, on a Saturday morning. You've seen light beams come into your house. Imagine your room is a leaf. Can you imagine a light beam coming into your room and as it being similar to a light beam coming into a leaf? What would you do if you were a plant? If you were designing a plant to capture that beam of light? How would you arrange your materials in order to capture them? To make sure you capture all the rays coming into the house, into the room. So think about it, and think about what enzymes are we talking about? We're talking about, ATP synthetase. You're talking about the cytochromes. You're talking about the chlorophyll pigments. You're talking about the whole photosystem 2, the whole photosystem 1. You're talking about the electron transporters, the electron carriers. You have to think about where they're positioned, how they're positioned. The other videos that I sent you also, I think, do a good job at describing that. So think about that. And again, this is a question that I really want to talk about in class. And certainly, again, all these questions are possible test questions. And your test, again, will be next Wednesday or Thursday. With regard to the proximity of the stroma to the thylakoids, why does light-dependent reactions happen so quickly after the light-dependent reactions? Well, this is true in C3 plants, but honestly, the C4 and CAM plants actually do things a little differently. Remember that, and we'll talk about that in class. But when you talk about the proximity of the stroma and the thylakoids, when we talk about how things are happening so quickly, well, the light reactions are making the ATP. The closer that the thylakoids are to the stroma, the quicker the NADPH and ATP from the light reactions can get to the enzymes in the Calvin cycle that produce the sugar. And remember that those spaces in the stroma allow for the carbon dioxide to diffuse into those spaces, and that carbon dioxide is a source of carbon for the sugar. So it all works out that the closer they are together, the faster those reactions can occur. How is the arrangement of thylakoids in the grana or stacks designed to maximize light capture? A lot of these questions almost repeat themselves. But if you think about stacking the thylakoids, it seems that it would make sense that would actually help in capturing rays of sunlight. All right, that's it. Sorry about my puppy. She's barking at somebody crossing the street. It seems no one can come near our house without someone getting barked at. All right, man. Ladies and gentlemen, have a great day. I hope this helped with your workbook. I'll send you one more video after this. Have a good one. Bye.